Assalamualaikum and good day everyone. Hi, I'm Dr. Iziana Kamal Bahrain from Biology Unit, Center of Foundation Studies for Agricultural Science, UPM. I'm going to talk about biology lab safety guidelines and lab report preparation. The safety rules and regulation are developed to help prevent accidents and to ensure your own safety and your fellow friends in the lab. Let me take you for a tour at Biology Lab as per. This is Asasi 1 building. Here there are 3 biology labs, 1, 2 and 3. Next is Bio Lab located at Asasi 7. Asasi 7 building is opposite to Asasi 1. Here there are 4 Bio Lab, 4, 5, 6 and 7. I would like to introduce 3 biolab staff that will assist all of you in conducting the experiment. They are Mrs. Sharina, Mr. Nazaran and Ms. Nurul Ain. When you enter the biolab, you must wear a lab coat and button up properly before performing the experiment. The lab coat provides an extra layer of protection against chemical spills and splashes. Second, you have to wear long pants that cover you to the ankle. Never wear shorts, skirts, and other clothing that does not cover your legs below the lab coat. Third, you have to wear shoes which completely cover the feet. Open-toe shoes, sandals, high heels shoes are not permitted to be worn in the lab. Glove and safety goggles are optional to be used in the lab. Before starting the experiment, there are 5 steps that should be done. First, you will have a lab manual that contains background and lab procedure to do the lab activities. Read it all and don't just scanning the manual with your eyes. In fact, you may need to read it more than once to get a good grasp to understand the lab manual. For this semester, we are going to have actual and virtual lab. The link of the lab briefing and visual lab will be provided later. Please make sure you have watched the video before entering the lab. I repeat, please make sure you have watched the video before entering the lab. This is to ensure that you are well prepared to conduct the experiment. To enhance your understanding on the lab procedure, you must write a summary of the methods or flow chart or answer the pre-lab questions. The instructor will determine what kind of pre-lab that you will have to prepare later. Before the lab started, normally the lab instructor will brief for 15 minutes about the experiment. If you have any inquiries regarding the experiment, please ask the instructor. I would like to emphasize there are 5 steps before starting the experiment. Read, watch, write, listen, and ask. This is an example of summary of method and flow chart of method. You may draw the apparatus and use arrow in the flow chart. Lab apparatus is essential tools in conducting an experiment. Examine all apparatus for defects, for example, cracks, before performing any experiment. Please handle all apparatus carefully. Try to avoid accident or breaking the apparatus. But if any glass apparatus is broken, please immediately report to the lab assistant so that we can record and update the apparatus inventory. Dear all students, please be extra careful with chemical handling. The prepared chemicals by the lab staff are placed specifically on a bench to be used by the student. Therefore, please do not move the chemicals from its original place. Always read bottle labels twice before you use the chemicals. Be certain the chemical that you want to use is the correct one. Use different droppers, pipettes for different chemical solution. Do not mix the droppers, pipettes to avoid chemical contamination. Do not take or use any chemicals more than is needed. If any chemical splashes into your eyes, please do not panic. What you have to do is rinse immediately with a large quantity of tap water. You should think before drain any chemical waste. Chemical waste should never be placed in a sink drain without permission. Please consult your instructor in all cases. After any experiments, make sure you have turned off all equipment and place it at a proper place. Next, do not leave any specimen. For example, onion sample that you have used should be thrown away into the bin. Wash your hand with soap and water after handling any chemicals and glassware to make sure your hand is free of chemicals. Clean up the bench and sink from any spill or stain. Wipe dry your bench with a clean towel. And lastly, do not leave any books, lab reports or personal belongings in the lab. What is a lab report? Have you written a lab report before? So a lab report is actually a formal record of experiment. So this record is very important to describe and analyze a lab experiment that you have done. This is also considered a basic training to produce a scientific paper for a scientist. A complete lab report is to be submitted in a group according to your instructor. Plagiarism or fabrication of data is forbidden in the scientific experiment, and strict action will be taken or your group marks will be demerit. So, how to write a lab report? A formal lab report must have 8 following components. The first one is the front cover. Second, objective or the purpose of the experiment. Third, materials and methodology. Fourth, results. Fifth, question. Sixth, discussion section. 7 and 8, conclusion and reference. This is an example of lab report front cover. It must have UPM logo, lab report number and title, group members' name and metric number. This is very important because we are going to give the marks according to the member's name and metric number. The last component is group members' task and signature. Please state the task that you have done in the experiment or lab report. Template of this front cover will be provided by the instructor. Next, let's move to the second page of lab report. Objective is a very important element in a lab report because you need to convey what is the purpose of your experiment to the reader. So it must begin with to. For example, to identify, to determine, to evaluate, and etc. Next is methodology. You should explain how an experiment was conducted. It is a step-by-step description of the procedure followed. Describe what you did and not how to do the experiment in the future. So you need to use past tense. You can also use diagrams. So you are encouraged to prepare a method of experiment in flow chart manner before coming to the lab. And then you can use this flow chart to be included in your lab report. Next part of lab report is results. This is where observations and raw data are recorded. You should organize the data obtained and do statistical analysis where applicable, depend on the experiment. Use table format or plot a graph where necessary. Scientific drawings, let's say for example from a microscope image, should be drawn with a clear labeling. There are several rules in scientific drawing. Look at the specimen carefully and examine the main features that will be included in the drawing. You only can draw what you can see. Let's say, for example, if you are observing an animal cheek cell under a microscope, the structures that you can see is only membrane, nucleus, and cytoplasm. Can you see ribosome or mitochondria under the light microscope? Definitely no. Therefore, you can't include mitochondria and ribosome in your drawing. Use pencil only and unlined paper in scientific drawing. Use solid single line, no shading or cross hatching. Only stippling is allowed. This form of shading is accomplished by simply putting dots on the figure. The closer together and more dots, the darker the part of the object will appear. Total magnification is a compulsory component to be written when you are drawing microscope image. Label only at one side, which is at the right side of the drawing. Avoid label at the top and bottom of the drawing. Next is rules of label lines in scientific drawing. Use a ruler to draw label lines. No freehand lines. Don't be lazy to use your ruler. Do not draw any fancy lines. Scientific drawing is not like your diary. Do not draw a label line crossing with other label lines as displayed here. Label lines should be pointed at the center of the structure. Untouched arrow like this is wrong. Do not use arrow at the end of the line. Do not write on the label lines. You should write the label at the end of the line. How many structures that you have to label? At least 3 labels per drawing. This slide shows a good example of biological drawing. As you can see from the drawing, it used single line, no shading, and only stippling, I mean the dots. It also used straight label lines, and all the labels are on the right hand side. At least 3 structures have been labeled here, and it also states the magnification. For the question section, you must answer all questions from the lab manual in your lab report. Marks will be given based on the answers. You are encouraged to find reference book to back up your answers. The discussion section carries the most weight in a lab report. Most educators pay the closest attention to this section because this is where students interpret what they have discovered and how well they understand what they have done in an experiment. So the student should be able to identify and explain the result that they have obtained in the experiment. Let's say, for example, you have observed plant tissue under a microscope. from the results, you can see the epidemics is tightly arranged. So you have to discuss why it is arranged in such a way. Next, you have to compare the experimental result that you have obtained with predictions, theories, facts from the reference. If it meets your expectation, so you don't have any problem to discuss. But if you get unexpected results, so you have to explain it. This is also where you would discuss any mistake that you have done while conducting the experiment. So you have to identify any sources of error or problem that might impact on the interpretations of your results. And you may also wish to describe ways the experiment might have been improved. The last section of a lab report is conclusions and reference. You have to ask yourself whether the objective of the experiment has been achieved or not, because this is a guideline to write a conclusion. The conclusion must be written in brief, accurate, and compact. Let's say, for example, you have 3 objectives, so you should have 3 conclusions. For instance, the objective of an experiment is to identify the important factors that affect the photosynthesis rate. So, in conclusion section, you have to state what are the identified factors that affect the photosynthesis rate. The best way to state the conclusion for a lab report is 1 or 2 sentences per objective. You also have to state the significant quantitative value. For example, state the highest rate of photosynthesis by using numbered value instead of just mentioning highest, lowest. For the reference, you have to state at least 3 references in APA style format. You may refer to this link on how to write the reference. Wikipedia reference is not allowed to be used as a reference in a lab report. And as a reminder, please submit the lab report on time, depending on the educator's instruction. For our first lab of this semester, we are going to learn the parts and function of microscope through Visual Lab. Please click the link in the description box and do the interactive activity accordingly. Further instructions and assignment will be given by your instructors. That's all from me. Thank you very much everyone for listening to this briefing video. Enjoy doing your experiment at Biolab. Stay safe and take care.